Hello students, so today the topic which we are going to discuss, that is cell wall, and you all know that the cell wall is the outermost boundary in case of prokaryotes, in case of plants, in case of fungi, but in animals the outermost boundary is the plasma membrane. So let me start the cell wall. Okay, in this class I will tell you about the different composition of the cell wall in different organisms. Okay, so let's say start cell wall. You know that the cell wall was discovered by whom? It was discovered by Robert Hooke. Okay, and cell wall is the unique feature of plants. Cell wall is the unique feature of plants. Who told this? Swan. Okay, and you all know that the cell wall is not present in animals, so I can say that the cell wall is absent in animals. Okay, now I am going to tell you about the different composition in different organisms. So let me start first bacteria. So in case of bacteria, the cell wall is made up of peptidoglycan. It is made up of peptidoglycan. You will ask me, sir, in all the bacteria? No. In most of the bacteria it is made up of peptidoglycan. Here the glycan part is the carbohydrate part, and it is having the two units. One is NAM and one is NAG. What is the full form of NAM? Actually NAM is N-acetyl-muramic acid, and what is NAG? NAG is N-acetyl-glucosamine. Okay, I am denoting NAM in this manner and NAG in this manner. So in the bacterial cell wall, this NAM and NAG are arranged in the alternate order, like this is NAM, this is NAG, NAM, NAG, then NAM, then NAG, then NAM. Like in this alternate order, they are arranged in the bacterial cell wall. Okay, and the bond which is present in these units, that is called as what? Beta 1,4 linkage. This is what? Beta 1,4 linkage. Okay, in the same manner another layer, this is NAG, NAM, then NAG, then NAM, then NAG, then NAM, then NAG, like that. Another layer, like this is NAM, this is NAG, this is NAM, this is NAG, this is NAM, this is NAG, like that. And these different layers are also interlinked by the another type of bonds over here. For example, this layer and this layer is linked like this way, and this is like this way. So these bonds which are interlinking the different layers, this bond is called as what, students? This is tetrapeptide link. This is tetrapeptide link. Okay, so we have understood one thing, that the bacterial cell wall is made up of the peptidoglycan. The glycan, the carbohydrate part, is having the two units, NAM and NAG, N-acetyl-muramic acid and N-acetyl-glucosamine, and they are arranged in the alternative manner. Okay, and the different layers are interlinked also with the tetrapeptide links. For example, sometimes you fall ill, you have any kind of the bacterial infection, so you go to the doctor and the doctor gives you antibiotics. What is the use of antibiotics? So antibiotics is actually doing what? That antibiotics either reduce the growth of the bacteria or it can kill the bacteria as well. Okay, how the antibiotics work on the cell wall of the bacteria? So let me give you an example. For example, we have one antibiotic, that is penicillin. How it works? So penicillin, you all know that the first discovered antibiotic, which was discovered by Alexander Fleming. So this is the first discovered antibiotic. How it works? Actually, this penicillin antibiotic break these tetrapeptide links. Okay, so when it break these tetrapeptide links, the cell wall of the bacteria get degraded and the bacteria ultimately dies. Okay, so one thing you have to remember here, that most of the bacterial cell wall is made up of peptidoglycan. This extra part I have told you, so you can remember that also. That is for extra knowledge. Okay, next one I am going to tell, that is in case of fungi. So in case of fungi, dear students, you have to remember that the fungi cell wall is made up of chitin, or you can call it fungal cellulose. chitin or fungal cellulose. Okay. So chitin is what here? Chitin is a polysaccharide. You know, chitin is a polysaccharide. In zoology also you have studied about the chitin, that in the arthropoda group, that the exoskeleton of the arthropods is made up of what? That is also made up of chitin. Okay. But one exception you have to remember here. What exception? That here one exception is present, that in the Oomycetes member, you will ask me, sir, what is Oomycetes? Oomycetes is actually basically the class of fungi. Okay, and exceptionally in the Oomycetes member, the cell wall is not made up of chitin, it is made up of cellulose. It is made up of cellulose. This is important, you have to remember. It is made up of cellulose. Now next, in case of plants. So plants case, we have to remember that the plant cell wall is made up of, that is cellulose. Next one, that is hemicellulose. Next pectin, and next protein. Okay, so these four components are present in the cell wall of plants. Okay, one more thing is given in your NCERT, that is algal cell wall composition. So for that algal cell wall you also have to remember that the algal cell wall is made up of cellulose. Next one galactans. Next one mannans, and next one minerals. And what type of minerals over there? Like calcium carbonate and SiO2, silicon dioxide. Okay, so this is the compositions of the cell wall in different organisms. Bacteria, the sole member of kingdom Monera, right? It is a prokaryote. Next one fungi, kingdom Mycota. Next one plants, kingdom Plantimember, and these are the composition of the cell wall in these different organisms. These basics you have to remember. Robert Hooke discovered, absent in animal cells, and this is the unique feature of the plants which was told by Swan in cell theory. Right, now I am going to discuss the next thing, types of cell wall. Next one we are going to discuss what? Types of cell wall. So now I am discussing next, that is types of cell wall. Types of cell wall. So to discuss about the types of the cell wall, I am taking the plant cell as a reference. For example, I am drawing one typical plant cell. Let's say this is the plant cell. So the first formed the cell wall in case of the plant cell, it will be what? It is the primary cell wall. So let's say this one is what? This is the primary cell wall. This is what? This is primary cell wall. Now this primary cell wall, you have some characteristics, yes or no? So the first characteristic of this primary cell wall, that it comes very first in the plant life. Okay, and it is the outermost covering. It is the outermost. So first thing is that it is outermost. Next, it is first formed. It is first formed cell wall. Okay, it is first formed cell wall. Okay, so this is outermost first formed cell wall. Next, that in all plants you will see the cell wall, so it is universal. Okay, next, as the cell matures, so this cell wall get diminished. So I can write that it diminishes as the cell matures. It diminishes as the cell mature. Okay, next characteristic is that the primary cell wall is thin. It is what? Thin. So I can write that it is thin. Next characteristic, it is flexible also. It is flexible, and you know that if it is thin and flexible, it is also capable of growth. So next one, it is capable capable of growth. Okay. So this is the first type of the cell wall, that is primary cell wall. Now I am going to draw the next cell wall, which is formed on the inner side of the cell beneath the primary cell wall. Let's say this one is what? This one is the secondary cell wall. This one is secondary cell wall on the inner side of the primary cell wall. So this one which I had drawn over here, this is secondary cell wall. Okay. And this one was what? This one was primary cell wall. Students, here you have to remember the composition of primary cell wall as well as the secondary cell wall. That both of the cell walls are made up of cellulose, next hemicellulose, next pectin and protein. So these four components will be there: cellulose, hemicellulose, pectin and protein. Okay. Now in this diagram you are noticing that the secondary cell wall is comparatively thick than the primary cell wall. Okay. So the characteristics of the secondary cell walls are that it is thick. Okay. Next, it is rigid also, and you know that if it is thick and rigid, it will have the different layers. Yes or no? So it is thick, rigid and multi-layered. It is thick, rigid and multi-layered. Okay. So in multi-layered condition, it will have the three layers: S1, S2 and S3. Okay. These three layers will be there in the secondary cell wall: S1, S2 and S3. Now you have to remember the thickness order also. Thickness order. So the thickness order of these layers will be S2 will be the thickest, then S1 and then will be S3. This thing you have to remember. This is the thickness order. Okay. So this is about the secondary cell wall. Okay. Now after the secondary cell wall, the next cell wall that is formed on the inner side of the secondary, that will be what? Tertiary cell wall. So this one I am drawing. This one is tertiary cell wall. It is tertiary cell wall. Okay. Now if I talk about the composition of the tertiary cell wall, so the composition is that it is made up of hemicellulose and xylene. Hemicellulose and xylene. So this is the composition of tertiary cell wall. So in this way we have discussed the different type of the cell wall, like first form, that is primary, the second one, that is secondary, and the last one, that is tertiary cell wall. Primary cell wall, these are the characteristics. Secondary cell wall, these are the characteristics: multi-layered, rigid, thick, and this is the composition of both of the cell wall: cellulose, hemicellulose, pectin and protein. Okay. So you have to remember the compositions. Okay. In the exam, that will be very important for you, that you remember all this composition of these different type of the cell walls. Now next I am going to discuss the two terms. One is plasmodesmata and one is middle lamella. Okay. So now the next topic is plasmodesmata. Next term is plasmodesmata, and one more term, that is middle lamella. Middle lamella. Okay. Actually, what is plasmodesmata? Plasmodesmata is basically the cytoplasmic connection between the two adjacent cells. That is plasmodesmata. And what is middle lamella? Middle lamella is the cementing layer which is joining the two cells, two adjacent cells. Yes or no? So let me explain with the help of a diagram. For example, I am drawing the two cells over here. Let's say this is cell number 1 and this is cell number 2. Okay. This is cell number 1 and this is cell number 2. I am not drawing the complete diagram. I am just writing the junction over there. So this is cell number 1 and this is cell number 2. And you know that the outermost cell wall is what? Primary cell wall. So this is the primary cell wall of this cell and this is the primary cell wall of this cell. Okay. Inside the primary cell wall, the next cell wall is there, that is secondary cell wall. So here this one is what? This one is secondary cell wall. Here also this one is secondary cell wall. So this is secondary cell wall of this cell and this is secondary cell wall of this cell. Okay. Now we have to make the connection between these two adjacent cells. So let's say I am making the connection between these two cells. So let's say here this is the connection between the two cells. Okay. And here let's say in this manner and here like this. This is what, students? This is basically the plasma membrane. This is the plasma membrane of this cell and this is the plasma membrane of this cell. Okay. And a connection is there between the two cells. Okay. Now the ER of this cell, ER, you know that what is ER? ER is the endoplasmic reticulum. And here this is the ER. And what type of ER is this? This is SCR. Okay. The ER of this cell will be in the connection with the ER of this cell. Okay. Or I can say the SCR of one cell will be in the connection with the SCR of the another cell. So here this cell and this cell both are in the connection. The smooth endoplasmic reticulum of this cell and the smooth endoplasmic reticulum of this cell both are in the connection. And this connection, students, is known as desmotubule. Desmotubule. In cell biology chapter, you also studied about this desmotubule. Okay. So this is desmotubule. Now understand one thing, that this is the primary cell wall, this is the secondary cell wall, this is plasma membrane, and this part is what, students? This part is what? This part is the cytoplasm. This part is what? Cytoplasm. You focus that this is the cytoplasm of this cell is in the continuity with the cytoplasm of this cell. So there is a cytoplasmic connection. Focus here, this is the cytoplasm of this cell is in the continuity with the cytoplasm of the another cell. And this cytoplasmic connection between the two cells is called as what? This is called as, this is what? This is plasmodesmata. This is plasmodesmata. Okay. And this is what actually? The cytoplasmic connection. This is the cytoplasmic connection between the two cells. Okay. Now you tell me one thing, that this cell is leaving, this cell is leaving. So this is leaving path or non-leaving path? Your answer will be what, sir? This is a leaving path. So this cytoplasmic connection, or you can say plasmodesmata, is actually a leaving path. That's why we can also call it, it is the symplastic pathway. Plasmodesmata is basically what? It is the symplastic pathway. Symplastic pathway. Two kind of the pathways are there. One is apoplastic and one is symplastic. So this one is what? Living pathway. That's why we are calling it symplastic pathway. Okay. So this is about the plasmodesmatal connection, how it is formed. Now you understand one thing, that this is the first cell, this is the second cell. In between the cell, both of the cell here are joined with this cementing layer. Okay. You know that when we join two bricks together, we use the cement over there. Yes or no? Cement and concrete and other things. And the two bricks are joined together. In the same manner, this cell and this cell are joined with the help of this cementing layer. And this cementing layer over here, this is called as what, students? This is called as middle lamella. And for the exam point of view, you have to remember that middle lamella is composed mainly of calcium pactate. Calcium pactate. Magnesium pactate is also present, but mainly composed of what? Mainly composed of calcium pactate. Okay. Here calcium will be there and pactin will be there. So mainly composed of calcium pactate. Students, you might have seen that when the fruit is not ripened, then it is solid and hard. What is the reason of this? Because of the intact middle lamella. But when the fruit ripens, then what happens? The middle lamella, this middle lamella get dissolved, and because of that, the fruit becomes what? Soft or ripen. In the plant growth and development chapter, you also have studied that ethylene is also responsible for the ripening of the fruits. Yes or no? So what happens actually, this middle lamella between the adjacent cell get dissolved, and because of that dissolution the fruit get softer and get ripened. Yes or no? Now you will see that this is first cell and this is second cell. Here also cell will be there, and here also, here many cells are there. I have just taken the two cells and I just explained how the where is the plasmodesmata and how the middle lamella is present in the cells. Okay, so this is about plasmodesmata and middle lamella. Okay, now I am going to discuss the next topic, that is plasma membrane. Okay, now the next topic that I am taking, that is plasma membrane. Plasma membrane. So dear students, for plasma membrane is present in the animals as a delimiting boundary. You all know. Plasma membrane, the most widely accepted model of plasma membrane was given by Singer and Nicholson in 1972. Okay, actually he used RBC, and you know that RBC is the red blood corpuscle. It is not having the nucleus and it is easy for the study. So the RBC was used to understand the structure of the plasma membrane, and the technique which was used, it was freeze and fracture technique. It means you first freeze the membrane and then fracture it into different components. So the plasma membranes most widely accepted model was given by Singer and Nicholson in 1972. So what is the components of the plasma membrane? What is present inside the plasma membrane, and what is the kind of nature of the plasma membrane? So the nature of the plasma membrane is quasi-fluid in nature. You will ask me that, sir, what is the meaning of quasi-fluid? Quasi-fluid means it is neither solid nor liquid, like the honey you might have seen. So like that nature, quasi-fluid in nature. Okay. So in the plasma membrane, if I talk, so plasma membrane is basically having the three things. The first thing that you will see over there, that is protein. First one will be what? Protein. And the protein will be of two types. One is extrinsic protein. Or you can call it peripheral protein. Okay. These extrinsic protein or peripheral protein are present on the surface of the membrane. The next type of the protein will be integral protein. Or you can call it intrinsic protein. This type of the protein is present inside the membrane. Okay. Now the two conditions are there, that either the proteins are completely embedded in the membrane or partially embedded in the membrane. On the behalf of that, these integral or intrinsic proteins are further two types. Either it may be totally buried or it may be partially buried. Okay, so the first component will be what? It will be protein. Okay, where the protein is present, where the other components are present, when we will draw the structure, you can understand easily. So first I am telling you the components of plasma membrane. So first one is protein, and protein is also two type: extrinsic protein, which is peripheral protein, this intrinsic protein or integral protein. It may be the totally buried in the membrane or it may be the partially buried in the membrane. Okay, the next component of the membrane will be what? Next will be carbohydrate. Okay. So for carbohydrate, you have to remember that carbohydrate is absent on cytosolic side. On the cytoplasm side, the carbohydrate part will be absent. Okay. The next one will be what? It will be lipid part. And in lipid, here mainly which lipid will be there? Mainly will be phospholipid. Mainly phospholipid. Okay. The main lipid which will be present in the membrane will be phospholipid. Okay. Actually the cell membrane is phospholipid bilayer. Okay. Two layers, one on the another, like in the sandwich you might have seen. In that manner, it's a phospholipid bilayer, and in that phospholipid bilayer, these proteins will be what? Embedded in that. Okay. So what is the main lipid over there? Mainly phospholipid. Okay. The next type of the lipids will be there. It will be sterols. Okay. Now in sterols, if plant cell is there, then phytosterol will be there. If animal cell is there, then cholesterol will be there. And if fungi is there, then ergosterol will be there. What is that? Ergosterol. Okay. And in case of bacteria, sterol-like compound will be there, that is called as hopenoids in case of bacteria. But mainly we are studying plants, animals and the fungi. In case of the plant, phytosterol, animals cholesterol, and fungi ergosterols. So these sterols will be there. What will be the use of sterols? We'll discuss in the structure also, but now I am also telling that the sterols provide stability to the membrane. Okay. They also act as a temperature buffer. Okay. You know that in the winter season, the membrane, the chances are there the membrane may freeze. So they increase the fluidity of the membrane. Okay. So this is the function of sterols. In animal case, you have to remember importantly, that is cholesterol. Okay. The next will be what? Next kind of the lipid will be, it will be glycolipid. So these kind of the lipids will be there. Okay. So this is the outline of the plasma membrane, that what kind of the components will be there. Okay. The plasma membrane, first one protein, then next one carbohydrate, and next one our lipid. So these components will be there. Now I am going to draw the structure of the plasma membrane, then you will understand that how these proteins, carbohydrate and lipids are arranged in the cell membrane. Okay. So now I am going to draw the structure of plasma membrane. So to understand the structure of plasma membrane, one molecule I told that it is present in the most abundant quantity, that is phospholipid, and I told that the membrane is a phospholipid bilayer. So to understand the first, I am drawing one phospholipid molecule over here. So this is one phospholipid molecule over here. This is phospholipid molecule. Okay. This is one phospholipid molecule. This part is what? This part is called as head, and this part is called as what? This part is called as tail. The head part you are seeing over here, the head part is actually, it is hydrophilic. What is the meaning of hydrophilic? It means water attracting. It means this part, head part, want to tend to stay towards the water content side. Yes or no? The next, this one is tail. This tail is what, students? This is hydrophobic. And what is the meaning of hydrophobic? This is water repelling. It means this part want to stay away from water, and this part want to stay towards water. Okay. In this tail, so also the two kind of the tails are there. This one is what? Saturated. This is saturated tail, and this one is what? This is unsaturated tail. Okay. So in the phospholipid molecule, the one tail is saturated and one tail is unsaturated. You might be noticing that here this tail is little bit bending, and what is the reason of this? Because in unsaturated hydrocarbons the double and triple bonds are there. Because of that bond angle, this here some bending over there, this is called as what? Kinking. So here kinking is also there. Okay, and in these two tails, saturated and unsaturated, the flexion movement is continuously going on. Okay, so this is the phospholipid molecule, head part, tail part. The nature of the head part is tends towards the water side,